Wait, is this like the paper that just came out in Nature, right? Okay, we actually journal club this. No, no, it was very good. Like it's not in my lab, but I'm co-supervising a student who works in a hippocampus lab, so she presented it. Yeah, it was like this is why the title sounded so familiar and your name. I hope so. Do you want to try?
Test.
Okay. Hello, everyone. Okay. Hello, everyone. Welcome to the 5th session of this exciting conference. And our first speaker is Weinan Sun from Cornell University. So, Weinan, the floor is yours.
All right. Thank you. So my name is Weinan, and I'm an assistant professor at Cornell University's Neurobiology and Behavior Department. So today I'm going to tell you about a recently published work that was done in HHMI Geneva Research Campus with the colleagues shown below. So just imagine you are a small mammal that is maybe a mouse roaming around in the grass. And then your goal is try to find food, avoid predators, and remember how to get back to your habitat. So it's really crucial for you to have the ability to form a mental model or so-called cognitive map of your surroundings for navigation and planning. So for humans, I think the ability is similarly crucial. You are just here in this meeting for only 2 days. And I can imagine that you already formed some form of preliminary cognitive map of this hotel. So I'm going to actually test you guys on that. So I want you guys to close your eyes and imagine you need to go to your hotel room. I'm going to do this myself too. Just imagine a path that how are you going to walk out from the conference room all the way to your hotel for a few seconds. So I'm sure many of you can somehow do that to some extent. And maybe some of you imagine that you're going to step out of this room and you're going to go across the hallway with the posters, and then you're going to walk to the elevators. So I think that is an indication that there's some kind of mental model present in your mind. And I was trying to prepare this presentation. I took the pictures in the hallway and noticed something peculiar about our world. So just notice that there are just a lot of repeats. You see the doors are repeating, the elevator doors also, and there are repeating lamps. There's also even like repeating carpet patterns. So, and that's interesting. So I took the elevator and go upstairs, and I intentionally step out on 2 floors. And then I turned east and turned west and took a picture on both floors, 2 hallways. And it turns out that they're extremely similar. So my question is, as animals and humans, how do we build a cognitive map? And especially in a world that we have a lot of perceptual ambiguity. In a patch of grass, many patches of grasses look the same, and the different floors in the hotel rooms looks the same. So to do that, we train mice to play virtual reality games. While doing that, we also image their brain activity, specifically the hippocampal activity, while they learn the game from day 1. So we use a microscope that was developed at Genelia called 2P RAM mesoscope, which has a large field of view around 5 millimeters. We use this microscope to image the dorsal CA1 activity from mice. This is an example animal. You can see that the animal is performing a virtual reality task, and we are simultaneously recording up to 10,000 CA1 pyramidal cells. So let me explain the task. The animals will randomly see one of the 2 linear mazes. So it's linear, and the walls are decorated by one of the 2 patterns shown here. The first one is called the near trial, and the second one is called the far trial. So the task is simple, that depends on the initial indicator, the identity of the indicator, the animal needs to lick either in the near location or the far location to receive a water reward. And one specific design principle of this task is that you can see that other than the indicator region, the rest of the track looks identical among these 2 trial types. So there are a lot of perceptual ambiguity. Here is a well-trained animal rendering on a VR screen. So you can see this is a far trial. It skips the first cue and then starts to lick here at the far reward zone. After licking, it enjoys the water and then run to the end. So there's a 2-second teleportation period connecting to the randomly selected next trial. So this trial is a near trial. So the animal will lick at the near zone, and if it is fully learned the task, it's going to skip the far reward cue, which it was licking on the other trial type. So you can see the animal can learn this roughly within 7 to 14 days. So I'm going to show you an example behavior from one example animal. On this plot, on the x-axis, I'm showing you the location along the 2 trial types. On the y-axis, I'm showing you all the hundreds of trials the animal went through across 9 days, specifically for this animal. The dashed lines separating 1-hour sessions between days. So as you can see that these animals were water deprived, and they are eager to get water. So on the first day, it's the first time they've ever seen this VR task in their life. They run, they lick all the time randomly. Only after a single day of learning, they learn something new. They learn that the first part of the track is never rewarding. So they stopped licking there and only lick at the potential reward zones only. Then something interesting happened. They learned an interesting intermediate strategy that they will run, not licking anywhere, and start to lick at the near reward zone. If they get water in the near trial, they will not lick and run all the way to the end, as if they learned something new that, oh, I know there's only one drop of water. If you get water, don't bother even licking again. However, that strategy doesn't work in the far trial. It starts to lick at the near reward queue. It doesn't get water. It will sustain a bout of licking until it gets water, and then it will run all the way to the end. So this is more efficient than licking at both reward queues, but still the animal doesn't understand the indicator queues deterministically determine the reward locations in both trial types. The animal were stuck in this phase for a while, and then gradually they start to appreciate the true underlying task rule. Eventually, they will only lick the correct reward zones in both trial types. So while the animal were performing these VR behaviors, we also record their neural activity in CA1 from day one. We process each day's calcium imaging movie, extract the ROIs, and extract the calcium activity traces using SueYeon developed by Carsen Stringer and Marius Pacutario at Geneva. So here is an example calcium traces from 60 cells, and you can see the animal is performing the task, and we are gradually adding more and more cells into the visualization. And as you can see that in the end, with all the cells shown, it's quite a large data set. It's hard to make sense of. We've done various analysis and tried different techniques. But for today's talk, I'm going to show you one of the more intuitive results. We used UMAP to do dimensionality reduction. We take each calcium imaging frame and reduce that from 5,000 dimension to 3. So we have 5,000 cells, and we want to reduce the temporal dynamics into the 3D space. We concatenate all the days of data, and we do a one big UMAP for all the data. So next, I'm going to show you one representative animal's manifold, the task manifold over learning. Here, I'm showing you the UMAP for session 1. Remember that we did all the day's data. We're only showing you this day's data and masking all the UMAPs of the other days. So here, each dot represents a single calcium imaging frame. The warmer color indicates the near trial. The cold color indicates the far trial. You can see on the first day, the animal knows nothing about the task almost, and is kind of an unstructured ball. On the second session, something interesting happened. It learned a preliminary structure of the task. And it's quite surprising to me that I thought the animal were going to learn the linearness of the task very fast because nothing could be simpler than a linear track. But I was wrong. It turns out the animal learns a simple transition between the gray queue. Remember, there are 4 gray queues interspersing the unique queues. The animal is treating the gray queue as the home state. It sees the gray queue, sees another queue, the brain state will come out, and it'll see the gray queue again. And it thinks it has come back to the same gray queue state. So the initial cognitive map is this hub and spoke structure. And it suggests that the animal doesn't even have the sense of linear track yet. And actually, the first learning step is to build this sequential task structures cognitive map. As you can see that after 1 day of learning, this animal disentangled the middle center gray queues and dispersed them along a ring at different locations, meaning that they start to appreciate that the gray queues indicate different locations. The black is the teleportation region that connects the end of the previous trial to the start of the next trial. As you can see, as the animal runs, it sees initially the stripes or the 4 dots. These are the embeddings for the indicator queue. However, at this early stage, after they run past the indicator cue, the two trajectories join back together again. It suggests that although the brain registers the difference between the two indicators, it's not using this information to guide future decision-making. The animal here licks at the first reward cue in this pink color in the near trial. It gets a big drop of water, and then it will stop, often stop, and then pause for a long time. And during this time, the embedding starts to show this random scatter at the off-manifold space. And we are not going to analyze these activities. We think it's an interesting form of memory reactivation. But ignore that for now for this talk. And after the animal starts to run, it will run back to the beginning, and then if the next trial is a far trial, it licks at the near reward cue. It doesn't get water. It keeps running. Licks at the far cue. It gets a big drop of water. And then the brain goes into this scatter state. Then it starts to run again. So on day 3, the animal will just running on this ring. The brain activity will be constricted in this ring over and over again for hundreds of trials. Then, the next stage of learning, something else happened. There's a split at the later part of the track, indicating that state that they start to lick at the near cue and then stop licking once they get a reward. Basically, they're gathering evidence at the near cue. They lick. If they get a reward, their brain state will go to one branch. That means that they should not lick. If they don't, their brain state will be in the other branch, meaning that they should keep licking until the end. And then the last learning stage is, of course, the expert animal start to build a split shank ring. After the indicator region, this crucial region that's right after the indicator start to bifurcate. And then the two trajectories will completely bifurcated until the end of the track. So, yeah, the animal have a stereotypical process of building this cognitive map. We performed computational modeling to explain this process, and to save time, the gist of the computational modeling is that we found one model that was based on a hidden Markov model called a clone structure causal graph. This model not only replicated the correlation structure between near and far trials across learning in the animal, you can see that in the end, it's a quite orthogonalized representation. But also, this model replicated the exact learning trajectory of the animal. To summarize, we found hippocampus developed an orthogonalized representation of the task. And we identified a systematic sequence of learning that the majority of the animals have that's correlating with animals' behavior. We also found that hidden state inference models like CSCG can capture hippocampal learning dynamics really well. And in the end, I want to conclude that we've also seen many single cell property changes over learning, like play cells and splitter cells. And we think that they can be generally understood as not plays or splitter, but as state cells that's representing the inferred current state. With that, I want to thank my collaborators at Genelia and Cornell. Especially, I want to point out that Jason Kim and Ida Cohen we're collaborating in developing a very quantitative manifold learning technique. So please encourage you guys to talk to them about that. Thank you so much.
Thank you, Weinan, for this really nice talk. We have time for a quick question while the next speaker sets up. Yeah, over there.
Hi. I was wondering if you tried out two-dimensional environments also to see whether you get place cell representation.
We haven't tried that yet. It's just, for now, only VR environments.
After this in the social. Our next speaker is Xulu Sun from UCSF, and yeah, Xulu, the floor is yours.
Thanks for the introduction. So the last talk set up well the idea of the cognitive map, and here I'm gonna extend on that idea, but more focusing on the value-guided decision-making. This work is done in Loren Frank's lab at UCSF. We know that foraging decisions depend on various factors, including preference, effort, and risks. The consideration of these factors form the basis of flexible decision-making. For instance, I've been debating in the past couple of days where to get the famous Montreal bagels for breakfast. If I had plenty of time before the first dock, I would go out to try the recommended shops, but if my schedule's tight, I'd grab something quick, although less fancy, in the hotel. Knowing these relevant factors and using them to predict and decide rely on our internal models of the world or cognitive maps learned from past experience. So here we try to investigate the neural basis of the flexible decision-making process. And we tried to create an analogous situation under the context of rat foraging. We designed a spatial banded task using the stem leaf maze. Two leaves in the same stem form a foraging patch, and at the end of each leaf is a reward port that delivers milk according to different probabilities that we change from session to session. There are two main rules in this task. First, rats are required to visit a different leaf site on each trial. And second, rats can choose between staying in the current patch or switching to another patch, and the reward probability of a given patch would be discounted if rats stay in the current patch, but reset to the original value if they switch to another patch. Here's an example of this discounting process when rats keep visiting the two blue sites. So first of all, do rats make reward-guided decisions in this task? Here I'm showing you the behavior of an example rat in the late learning session. In this session, the blue patch has the highest reward probability, followed by the yellow patch. Here, each circle indicates which leaf site the rat visited on each trial and whether they got rewarded. Usually, after initial exploration, rats would quickly hone in on the best patch, and in many cases, they also often later tend to stay short in the less rewarding patch before returning to the best patch, which means that the rats learned the depleting and resetting structure of rewards in this task. One important decision-relevant variable that we don't have direct access to here is the value of options. So we first need a way to infer the internal valuation of options before looking into their neural basis. To do so, our collaborators Ari and Nathaniel at Princeton University developed a hidden Markov model to infer the hidden states, which are the joint reward probabilities of all leaf sites on each trial, and using the observations of the reward outcome on each trial at a visited leaf. This HMM not only allows us to infer the internal value of all 6 sites on each trial to study the neural basis, but it also learned the depleting of local rewards and the resetting of remote rewards from rat behavior, which suggests that the values of both remote and local patches are critical for rat decisions. So here we define one decision-relevant variable, the relative switch state value, as the difference between the maximum value of remote patches compared to the value of the upcoming local leaf. Next, we want to understand how the brain may implement this value-guided decision-making process that could utilize an internal model of the world. Concretely, we wanted to address the following questions. How do single neural activity and population dynamics co-encode multiple task-relevant variables? And do representations of these variables enable the system to predict upcoming task states and decisions? Specifically, we focus on the prefrontal cortex that's been thought to be critical for encoding cognitive maps and guiding flexible decisions. First, to examine how these PFC neurons encode the task structure, we divided all state trials into these 6 journeys. And here's the activity of an example neuron across these journeys. One primarily encoded variable in many PFC neurons like this one is path progression, which means that their temporal firing patterns don't depend on specific journeys, but instead reflect in general how far the rats are from the starting point, the decision point, and the goal of each trial. On top of such task structure coding, do these PFC neurons also encode the switch state value? So we further divided trials of each journey into 4 value groups. And then we found that many PFC neurons are modulated by the switch state value. Here, this same example neuron shows consistent and generalized value coding patterns across all 6 journeys. The second example neuron here shows distinct path progression coding for journeys sharing left turn actions versus right turn actions. And its value modulation also depends on both path progression and action. across the PFC population we recorded from, we've seen this kind of multiplicative coding of value and task structure in many cells. And beyond these two examples, we've also seen various ways of how the value coding can be mixed with the task structure coding across different levels of condition generality. Given such diverse single neural mixed coding patterns, we wanted to understand how the neural population encodes value in conjunction with path progression, action, and patch identity. So we first turned to PCA. We grouped all trials by how far they are from a switch decision, and then applied PCA to neural data during the runtime of each trial. We found that largely along PCs 1 and 2, there are such rotational dynamics reflecting path progression. And here, this dark trajectory traces out neurodynamics on trials far from a switch decision. Then, across para-switch trials leading up to a switch decision, again, we saw the progression-related rotational dynamics largely dominating PCs 1 and 2. And we've also seen this orderly arrangement of the neural trajectories reflecting the increasing tendency to switch. And finally, on the switch trial, this yellow neural trajectory shifts further along this gradient. And if we plot the HMM estimated switch state values across the same para-switch trials, it becomes clear that the arranging order of these neural trajectories actually correlates with the value estimates. Similarly, we've seen this kind of value-related gradient of the neural trajectories across all journeys sharing left turn actions, shown here in shades of red, and also across all journeys sharing right turn actions, shown here in shades of blue. We also saw this kind of dynamical motif for journeys in each foraging patch, and we think that such a regular dynamical motif across different levels of condition generality could allow the system to track path progression and value associated with various conditions. Now, to briefly summarize, here using a spatial foraging task, we found that individual PFC neurons are selected for path progression, action, and patch identity with multiplicative modulation by value. We identified structured population activity that shows rotational dynamics driven by path progression and displaced by value. We also saw similar dynamical motifs that coexist across different levels of condition generality. Then we wonder how representations of these variables might enable the system to predict upcoming task states and decisions. So we next tried to find neural population dynamics that could reflect a predictive model of the world. First, let's review the neural trajectories in the navigation subspace, and you may notice that the initial states are already separated by the time rats start the next trial, which means that we could potentially find reward update signal related to future trials when rats pause at reward wells. So we used PCA again and built another subspace, the outcome subspace, using neural data from 0.85 seconds before movement initiation to 0.25 seconds after. We then projected the full trial neural population activity into this outcome subspace and found a spiral dynamical motif across the para-switch trials. Here, when rats stay in a given patch for a couple of trials, you can see that the spiral neural trajectories gradually shifted rightward, reflecting the ramp up of the switch state value due to reward depletion. Then on the switch trial, the yellow neural trajectory shifted to the rightmost side of the spiral even before rats started moving, and then reset after rats switched to a new patch, but before they reached the reward port. This suggests that this spiral dynamics could predict upcoming task states before knowing the reward outcome. And remember that the behavior results indicate that rats learned the resetting of the rewards in this task. So could the reset of the neural trajectories mirror such an internal model of value? So next, we directly decoded the switch state value from the neural population activity. Specifically, we combined all 6 journeys and divided them into 8 path progression bins, and then we fit the value decoder separately for each bin. We were surprised to see that this value decoder could persistently explain 25 to 35% of the total variance of the switch state value inferred from the HMM. And you could see that the value decoding performance is also higher before the decision point, as one would expect for values that could inform upcoming decisions. As a side note, we were also able to decode this switch state value separately for left turn versus right turn journeys, and for journeys in each foraging patch, as well as for individual journeys. With this value decoder, we can now project the neural population activity onto the value coding axis to review whether there's any ramp up and then reset structure in the neural value representations. And indeed, we found that across pre-switch trials before a switch decision, the decoded switch state value gradually increases. And then on the switch trial, before rats reach the decision point, the decoded switch state value peaks, suggesting that rats have strongly devaluated the current patch. After rats switch to a new patch, but before they reach the reward port, the decoded switch state value drops dramatically, indicating some optimistic value prediction in this new patch, even before rats experience any reward outcome there. This is consistent with the reset of the spiral dynamics in the PC subspace. Together, these results indicate that instead of passively tracking features of the environment, PFC representations can actively predict upcoming task states as one would expect from a cognitive map. To piece information together, we also looked into how the value coding axis relates to the spiral trajectories in the PC subspace. So we projected the value decoding axis of progression bin 1 onto the top PCs of the outcome subspace. And we found that this axis largely aligns with the direction where we saw the shift of the spiral trajectories across peri-switch trials, consistent with the function of tracking and predicting value. And finally, if these neural value representations are indeed useful for decision making, we should be able to predict the single trial switch state decisions from them. So we use logistic regression to do that. And we found that both the sensitivity of the prediction, which predicts switch decisions, and the specificity of the prediction are well above chance level across all rats. Now, to sum up here, using a spatial bandit task with multiple foraging patches, we found that individual PFC neurons are selected for path progression, action, and patch identity with multiplicative modulation by value. We identified structured population activity that shows rotational dynamics driven by path progression and displaced by value. We also found that similar spiral dynamical motifs coexist across different levels of condition generality. And finally, the spiral motifs and the neural value representations both track task-relevant variables and predict upcoming task states to support flexible decision-making across various conditions. This work is in collaboration with Alison Comrie, who's also in the audience. If you missed her poster, please stay tuned for a workshop talk about our hippocampal findings. I'd like to thank all the co-authors, including our collaborators, and many thanks to other members of the Frank Lab, our technical support groups, and also funding sources. Thank you all for your attention, and I'm looking forward to your questions.
Thank you, Zulu. I have a quick question while people get excited about what they're thinking about. So you see these like trajectories separate in PFC, like depending on the direction that the rat is moving, as well as all the cognitive signals. Do you think that this is something that reflects the motor process, or is it a byproduct of the cognitive map that they develop?
Right. So we also were wondering if it's like a motor-related signal, but I would say potentially the action-related signal could be something either motor-y or like right upstream of motor-related signal. But beyond that, it's kind of more like about the abstract task structure. Because you could also see, which I didn't show today, you could also see value-related signal for each individual foraging patch. Or you could predict which foraging patch the animal is in beyond like motor signals. But yeah, I do think there's like a mixture of all of them.
Okay, we can chat later. Yeah, like there's a quick question and answer from there.
Great talk.
Thanks.
I thought the reset behavior was kind of interesting. To what extent did you see variation in resets? And did you ever see an imperfect reset?
Right, that's a great question. So let's see if I have this. I have something in the, sorry. There's this variation of reset actually across days. What I showed you previously was just that if I group all trials by how far they are from a switch decision, there's a reset after this switch. But here I'm also showing you, they are like leaving the best patch and going to the medium patch versus leaving the medium patch versus coming back to the best patch. And in later learning days, the rats actually have different levels of reset for those two patches, which means that the rat actually predicts that the best patch has a better reward outcome if they switch there versus if they switch to the medium patch. So they not only learned that there's reset, they also learned that there's a different level of reset across the patches.
Okay. Thank you, Xulu.
Thanks.
Let's thank Xulu again. And our next speaker is Yue Liu from NYU. So Yue, the floor is yours.
All right, so thank you very much for this opportunity to share our work on the neural mechanism of hierarchical decision making, which is actually share many similarities with the paradigm that Xulu just talked about. So decisions in real life are often guided by some context. For example, in science, we design experiments based on some hypothesis. And when the outcome of the experiment is consistent with the hypothesis, it would strengthen it. But when it's inconsistent with the hypothesis, it can be caused by different reasons. For example, it could be due to some experimental error, or it could be that the hypothesis is wrong. So these two causes can be disambiguated using the confidence about the outcome of the experiment. So if we're new to the field, we would have lower confidence and probably go back and redo the experiment. On the other hand, if we're experts with a high confidence about the experimental result, we would tend to think that the hypothesis is wrong. And furthermore, if the outcome of future experiments are again inconsistent with the hypothesis, then it is more likely that the hypothesis needs to be changed. Whereas if they're consistent with the hypothesis, then the previous inconsistency is likely due to some experimental error. So in general, our decisions are guided by some context, and then we can use the outcome and confidence across a sequence of decisions to update the context. And this can also be thought of as a decision-making process on a longer timescale across several trials. So to formally study this process, we designed a hierarchical decision-making task. During each trial, monkeys need to discriminate the motion direction of a random dot stimulus, and the difficulty of each trial is varied by changing the fraction of coherently moving dots, which we call motion coherence or motion strength. At the same time, there's a hidden environment that changes stochastically across trials. So if the monkeys are in the correct environment, they can get reward by making a saccadic eye movement to the red target that corresponds to the correct motion direction. On the other hand, when they're in the wrong environment, all directional choices will lead to negative feedback, and they need to choose this blue target to switch to the other environment in order to get reward. And when they choose the switch target, the arrangement of the targets on the screen will change in the next trial to remind them about the current environment that they're in. Specifically, the target for leftward motion remains the same across environments, whereas the locations for the rightward target and switch target are swapped. As I will mention later, this is designed in order to separate the encoding of the saccade direction and other task variables. So here's the monkey's performance for example sequence of trials. We see that when the environment changes, monkeys also choose the switch target after experiencing some error trials. For example, here, the monkey switches environment after receiving one error, and here it switches after receiving 2 consecutive errors, and here it switches the environment incorrectly. So overall, we found that the monkey's accuracy for motion direction improves with motion strength, which is expected. And following positive feedback, they almost never switch environment, which indicates that they understand the persistence of the environment. Following negative feedback, monkeys do switch environment, and the probability of switching depends on the motion strength. For more confident trials with a higher motion strength, they switch right away. For less confident trials, they are less likely to switch environments. So this indicates that not only negative feedback, but also their confidence drives the switching. And furthermore, it does not only depend on the last trial, because when they experience a sequence of negative feedbacks, the switch probability increases. So this is similar to accumulation of evidence towards a bound, but on a longer time scale. So we refer to this evidence as the switch evidence. So from these results, we can conclude that monkeys perform decision-making both within the trial for the motion direction and the cross trials for environment switching. And the outcome and confidence of the motion decision informs the switch decision. And in fact, previous studies have shown that this kind of behavior can be described by a normative model that uses Bayes' rule to update this, to integrate the switch evidence. In this case, the switch evidence is proportional to the logarithm of the inverse error rate. So to understand the neural mechanism behind this behavior, we simultaneously recorded from 3 areas: the lateral intraparietal cortex, which is implicated for short-term integration of sensory evidence in direction discrimination, the supplemental eye field, which is implicated in error monitoring, and the dorsolateral prefrontal cortex, which is implicated in representation of abstract rules. And neurons in these 3 areas the neurons recorded in these 3 areas have response fields that overlap with one of the 3 targets. We found, however, that neurons in all 3 areas represent all components of the task. So here's a neuron in LIP that unsurprisingly represents the saccade direction. Here's another neuron in LIP that represents the environment. So here we're only focusing on the leftward saccade trials. So the difference in firing rate here is related to the difference in the environment. And here's another neuron in LIP that represents the switch choice. So here we're balancing the number of upper right saccade trials and lower right saccade trials. So the difference in activity is not due to the saccade direction. So we see that neurons in LIP represent direction, environment, and switch decision. And it's also the case for the other 2 brain areas, SEF and DLPFC. On the population level, all the task variables can be decoded in all areas. So here's the decoding accuracy for motion direction in LIP. You can see the decoding accuracy starts to rise after the stimulus is presented. And a similar time course was found for SEF with a faster rise time, and a similar result for the DLPFC. And we can also decode the environment in all 3 regions. In particular, we can see that the decoding accuracy is already above chance before the target onset, which means that the population represents the environment in a persistent manner. And we can also decode the switch choice even before the trial onset, which means that the neurons represent the switch decision variable also in a persistent manner. So we wanted to further understand the structure underlying this distributed representation, and in order to explore potential mechanistic hypotheses and inform data analysis, we trained a recurrent neural network to perform this task. So the network receives inputs about the outcome of the previous trial, the subjective environment of the previous trial, and the sensory stimulus, and was trained to output the saccade direction. In order to produce realistic behavior, the network was trained to mimic this normative Bayesian ideal observer model that I just mentioned. And after training, we found that the network can produce the same properties in the behavior of the monkeys. In particular, it is also more likely to switch environment after making an error for an easier trial, and also after making more consecutive errors. So next, we looked at the dynamics of the network and compared it to the neural data. First, we projected the network activity down to a low-dimensional space, where the axes of this low-dimensional space were found by using linear regression to predict different task variables from the population activity, and then orthogonalizing the weight vectors from linear regression. So here we can see that during the trial, when the network integrates motion evidence, the network integrates the motion evidence by moving horizontally, and the amount of horizontal movement represents the total amount of motion evidence accumulated, which we call motion decision variable, DVM. So this is shown for the post-correct trials, where the network does not need to switch environments. And for post-error trials, where the network has some tendency to switch environments, we see that the dynamics are similar, but interestingly, the neural states are translated along this direction into the screen. So we see that the network accumulates motion evidence by moving along this horizontal axis, but what does the movement along this other direction represent? So it turns out that if we color the same trajectories using the switch decision variable, trials more into the screen have a higher switch decision variable, which means that the network accumulates switch evidence across trials along this particular direction. So taken together, we found that the network accumulates the 2 types of evidence on 2 very different time scales along these orthogonal axes. And the same geometry was also present in the neural activity when we performed the same analysis. So So here I'm showing the result for one of the brain regions, but the result was similar for the other 2 brain regions as well. In addition, we found that these orthogonal directions are approximate line attractors. So a property of line attractor is that the network evolves slowly near the line attractor. Therefore, we identify line attractors by looking for these slow points where the network activity changes slowly in the absence of input. And we found that the direction that represents the switch decision variable has the most slow points. And these slow points also extend along the direction that represents the motion decision variable. And furthermore, there's a cluster of slow points near these post-correct trials where the switch decision variable is 0. So given these dynamical structures, we can start to see a mechanism that the network uses to perform this task. At the start of each trial, the network state is at some location along the switch line attractor, depending on the current switch decision variable. And then during the trial, the network integrates motion evidence by moving along this motion line attractor. At the end of the trial, if the network receives the negative feedback, it will be pushed away from the motion line attractor and fall back onto the switch line attractor. And this leads to a displacement along the switch line attractor at the start of the next trial. And this displacement represents the switch evidence caused by the error trial. On the other hand, if the network receives a positive feedback, it will be pushed back to the attractor that represents 0 switch decision variable. So I want to emphasize that this interaction between these line attractors only happens at the time of the feedback. So in this sense, it is gated by feedback. Now, for a more difficult trial with a lower motion strength, the network would move less along the motion line attractor. And if it gets an error, then based on what we know about the behavior, the displacement along the switch line attractor should also be smaller. In other words, the displacements along the 2 line attractors, D1 and D2, should be positively correlated. And indeed, we found this positive correlation in the network activity as well as in the neural data. So currently, we're still trying to understand what structure in the network causes this positive correlation between these 2 distances. So in summary, I showed that in a hierarchical decision-making task, monkeys accumulate 2 types of evidence on 2 distinct timescales: motion evidence within the trial and switch evidence across trials. And both monkeys and RNNs use orthogonal directions in the neural state space to integrate the 2 kinds of evidence. And furthermore, these orthogonal directions are approximate line attractors of the network. And although we cannot directly identify line attractors in the brain, we proposed a mechanism based on interacting line attractors. And this mechanism suggests a geometric signature in the population activity that we found in both the neural data and the model. And with that, I want to thank all the co-authors on this abstract, the Kiani, Wang, and Chung labs, as well as the funding sources. So thank you very much for your attention.
Thank you, Yue, for the really nice talk.
Cool talk. I have a question about the axis alignment in the neural data. So if an RNN isn't going to care if you have a really slow time scale in the same architecture as the fast time scale, but that may actually matter in brains. And so my question is it just kind of 2 line attractors in different parts of the brain? That is to say, the axis, I think it was 2 or 3, is neuron aligned or is not neuron aligned?
Yeah, so actually we performed some additional single neuron analysis that I didn't show here, which shows that neurons actually encode the conjunction of decision variables, both on the fast time scale and on the slow time scale. So in that sense, the decision axes are not neuron aligned.
Thank you.
Thank you. Really interesting talk. I just had a quick question about the axes again, but this is a question about how you orthogonalize the decoder weights, and I guess I think that maybe many task variables can have sort of shared subspaces and representations like this, and I'm wondering if you orthogonalize the axes, right, you lose a lot of this shared information that might have existed in a shared subspace, and then this maybe changes the dynamics. You have an intuition for why this why it's okay to orthogonalize?
Right. So yeah, you're right that this is probably not the most rigorous way of showing orthogonality. So here I'm showing the raw Cosyne similarity between the axes, and the boxed elements represents the cosine similarity, the angle between the two decision axes. And you can see they're close to 90 degrees. And this is the distribution across an ensemble of RNs. So they're also centered around 90 degrees.
Yeah, got it. Thank you.
Yeah,
Awesome.
We have time for one more quick question and answer while the next speaker sets up.
Yeah, thank you so much. That was a really interesting talk. And you might have just answered it with that previous slide. I'm wondering if you've separated out unsigned coherence or decision difficulty from the probability of switch. We've seen previously that difficulty signals and decision signals are orthogonal in these areas, and I'm wondering whether or not those two things can be decomposed with your analysis.
Sorry, could you say again which two signals are orthogonal?
Probability of switch and the unsigned coherence.
Yeah, so we do found that unsigned coherence correlates with probability of switching. But I guess I don't know how to do that neurally, because probability of switching is, I see what you mean. Yeah, I think actually these two axes are also quite orthogonal. The unsigned coherence axis and the DVS, which is the decision variable for switching axis, is also quite orthogonal. But it just so happens through the dynamics of the network that the larger the unsigned coherence, the higher the probability of switching is, or the larger the switch evidence is.
Awesome, thanks a lot.
Yeah, let's thank you again. And our last speaker of the day and last invited speaker of the day is Beth Buffalo from University of Washington. So Beth, the floor is yours when the tech helps us. Yeah.
Thank you. All right, so thank you to the organizers for inviting me. It's really great to be here. Thank you all for hanging around before the party. So it's hard always being the last speaker before the social. And I also, at the risk of taking us away from science for a minute, I also just want to acknowledge particularly the trainees, all the chaos that's happening across the world. And I know for a lot of trainees who are on visas, this is particularly anxiety producing when you're traveling. So I just want to say that we see you and acknowledge all the efforts that you've made to be here. And so now we'll go back to our Cosyne Island and get back to the science. And I'm really happy to talk with you about some new data, unpublished data that we have. And the title is Towards a Unified View of Hippocampal Function. It's really an aspirational title, so we're not there yet, but I do feel like the field has really been moving in this direction, and I think the talks earlier in this session even have really been also looking towards that kind of more unified function, or unified view. And so what do I mean? Like, what are we trying to unify? So probably everyone here knows kind of the two landmark discoveries that have really framed our understanding of hippocampal function. So work by Brenda Milner, which is also really poignant to point out since she did a lot of her work right here in Montreal. But what she did really back in the late 50s was pioneered the idea or understanding of the role of the hippocampus in memory with her studies with patient HM and others who suffered impairments in memory after having their hippocampus removed to relieve from epilepsy. And then there's this sort of parallel line of research that's been going on since the early 70s, pioneered by work from John O'Keefe and then Edvard and May-Britt Moser and others that have shown these really exquisite spatial representations. And so the field really for decades has been trying to reconcile these views to understand how it is that the same structure can be involved in both memory formation as well as spatial navigation. And so my work has really been trying to, for years, trying to help with this reconciliation by doing studies in the non-human primate, recording from the hippocampus, and trying to look at tasks that could be comparable to tasks that could be done in humans, where there's looking at memory formation. All right, so just to give you kind of a roadmap for what I'm going to talk about today. So in the first part, I'm going to describe some of our experiments using virtual reality. So these are essentially interactive video games in monkeys to try to understand the nature of the hippocampal cognitive map. And in the second part, I'll sort of shift and talk more about eye movements. And I'll describe what we've learned from studying activity aligned to these exploratory eye movements, these exploratory behaviors, which for primates really consist of saccadic eye movements primarily. And the overall take-home message that I'll try to leave you with today is that the hippocampal code comprises an internal model of a constructed sense of experience. So my hope is to sort of take us away from the idea that what the hippocampus is doing is reflecting a veridical representation of the outside world. But instead, the hippocampal code really reflects this kind of constructed sense of a mental model rather than a sort of veridical cognitive map. Okay. All right, so I'm going to start with a very old video. So some of you, if you've heard me talk, you've seen this before. So this video I love because it was one of our first ones using the video games. This monkey is Gromit, who you can tell got really good at using the joystick. And what he's doing is just navigating around in an open field. And his job is to collide with the banana. I don't know if the sound, if we can turn up the sound, but there it is. So what you'll hear is this beeping sound whenever he collides with the banana. And he gets actual food reward delivered. Oh, that's really loud here. And you can see he really shows these interesting behavioral strategies. So like that last one, he kind of went for the clustered bananas that he could get in one forward motion. They also tend to show that they can remember the location of the next closest banana, even when it goes off the screen. And he's learned a lot about how to do this task. So again, you can see how comfortable he is with the joystick moving around. And that white circle indicated where he was looking on the screen. So we can get lots of really rich information about behavior in these kinds of tasks. So of course, when we first started doing this and got our neural recordings in the hippocampus, the first question is, well, what do the place cells look like? Or are there place cells? And it turns out that we did not find any place cells. So what I'm showing you here is an example cell. As the monkey's moving around in the virtual arena, the red dots indicate where the animal was when the neuron fired. At the bottom is kind of a heat map representation of that. And then here's 6 other cells showing, again, a real lack of any kind of spatial selectivity. And while this was disappointing at first, we started thinking about it. And what's interesting is when you're watching your monkey or your animal perform the task, I think you can get a lot of intuition about what they're doing. And one thing that we realized when we were doing all these recordings is we actually weren't forming a very good map of that environment. There was no reason to really pay attention to those landmarks. All we had to do was look for the next closest banana. And so our thought was maybe that's actually what the monkey was doing, and maybe that's an explanation for why we didn't see any spatial representations. I just cited some other papers here from Rob Gooley from Julio Martinez Trujillo's lab and Dun Mao from Dora Angelaki's lab. They also have recorded in the hippocampus. Dun Mao was recording in a freely moving monkey and also didn't find lots of clear evidence of spatial selectivity among hippocampal neurons. So it's sort of consistent across the monkey studies. So we next decided, well, let's give the monkey a task where space matters and see if now we find some kind of spatial representation. So in this task, and this was work led by John Rookman and Yoni Browning in the lab, the monkey's job is to go down this center stem and then alternate turning left and right on different trials to find the banana now hidden in the tiki hut at the end. And if you can hear this sound, this is the sound you're hearing is the spiking of a single hippocampal neuron. And so it fired a little bit at the choice point, and then it fired again at the end of the leftward arm, but it didn't fire too much at the end of the rightward arm. So we'll play a couple more trials, and I'll show you. And I just want to call your attention, I'll be quiet there, so it didn't fire there. call your attention to the line at the bottom, that colored line that sort of shows the progression through the task, and that's going to be important in the coming slides. So you can hear that activity kind of there at the choice point. And then again, pretty robust at the end of the leftward arm. And so, we started seeing stuff that looked like potential spatial representations. And so what I'm showing you here are 9 different neurons, and they're split according to the leftward going trials and the rightward going trials for each neuron. At the bottom, you can see a kind of linearized version of this Y maze track, and it shows the heat map representation of the activity. And so hopefully what you can appreciate from this is that the neurons are pretty reliable. So each row is a different trial, and so you can see that you're getting a pretty reliable response. You could sort of decode activity or decode location from this activity. What you're probably also seeing is that they may not look like what you think place cells would look like, in the sense that they're not these sort of Gaussian punctate kind of responses. A lot of them are really tight, but others are very broad. And many neurons have multiple fields, multiple firing fields within the maze. And so as we started, looking at these again and watching the monkey performing the task and listening to the activity, we had the impression that these responses were really aligned to what was going on in the task, rather than exactly where the animal was. And so just to show you this now, I've added red lines that indicate the sort of boundaries between behavioral epochs. And what we found through further analysis is that all of these responses tend to be really well aligned if you think about the behavioral epochs rather than where the monkey is in space. So another way to look at this is if we take the whole population of neurons that showed robust and reliable activity in the task, and we've just sorted them now by their response latency. And so you can see, so each row is a different neuron. So you can see that this activity really spans the entire trial of the task. But you can also probably start to see kind of hints of shading at particular regions within this population activity. And so what we did then was look at the population vector and correlate the activity across different points in time. And just sort of for 2 extreme possible outcomes to give you a sense of what we might find. In the one case, if these were place cells that really represented unique locations in the maze, what we might expect to see is something like this diagonal, where you would see strong activity along the diagonal, but not much off the diagonal, because the population vector would be, again, representing unique locations. But by contrast, if the data or if the population really was coding more like a behavioral epoch, you might see something like this graph on the right, which is showing this sort of homogeneous coding across the behavioral epoch and then a sharp transition when you get to the next one. And this is what we found in the data. So this is showing the correlation in activity for a leftward going trial, for all the leftward going trials correlated against the leftward going trials. And so you can see these colors represent the different phases of the task. And hopefully what you can appreciate is that there's really this kind of block-like structure where you get, again, homogeneous kind of representation within a behavioral task phase, and then a sharp transition between the phases. And what's really critical about this is even in cases where the monkey is moving quite a bit, so down the center stem or moving along a sidearm, what you see is that the activity is kind of homogeneous within that, even though the monkey is transitioning quite a bit in space. And so, again, this suggests that it's really not about encoding the allocentric space, but it's something about sort of where the animal is in task space, and also that it's chunking it according to that. Here, the dotted lines just kind of give you that visual of where the boundaries are. Okay, since we were recording across populations now, we could also look at the activity and how it might be reflected onto within a multidimensional space, and thinking about the manifold that might exist there. So this is using PCA to identify a low-dimensional space for this population activity. And again, these colors represent the same task phases that are shown down here at the bottom. And so what we found is that there was a low-dimensional space that could be identified based on the population. But what was interesting is that if you look at, there were moments in this trajectory where the neural activity appeared to kind of hover in space and became almost static, even though, again, the monkey was moving through space. So at the population level, we also see this sort of warping of space relative to the behavioral relevance of the task. So John Ruckman, who's led this work, has also looked at the variability across trials. And so this is just a visualization of that with the variability represented by the size of these kind of bubbles, and the colors also map onto the task phases. And again, what he found was that the population activity remains this kind of almost static through conceptually similar task epochs, and there's this reduced variability at the event boundaries. So, our conclusion from this is that, again, what's happening in the hippocampus is this representation really is not what is sort of a veridical representation of the outside world, because we're getting this kind of shrinking and scaling of the world based on what's behaviorally relevant. And in an attempt to, again, kind of think about how this unifies across species and across different studies, this is something I think that is very consistent with work that we've seen for many decades in the rodent. So even from early on, we knew that place cells that showed direction-specific tuning. So these are some data showing that the neural activity in the rodent hippocampus shows a very different response when the animal is going inward in this maze versus when they're moving outward, even in the same allocentric place. And there's also been lots of studies showing that there's behaviorally relevant clustering of place fields, so that the map in the hippocampus isn't really continuous or homogeneous, but it shows this kind of chunking based on what's behaviorally relevant. All right. And so again, I think this all sort of leads to the idea that for the hippocampal code, it's really about this internal model of a constructed sense of experience. And so if that's true, then what we'd really like to do is see how that sort of map, that construction of the map or the internal model, while the animal is learning something new. And I think this is really sort of nice to follow the work by Weinan, who I think showed something similar in the rodent, kind of showed how that map gets constructed through learning. And so in my lab, this is work that was led by Sophie Landy, who's a really talented postdoc, and she decided to look at behaviorally relevant representations and how they evolve through learning by training the monkeys to perform two different tasks, one that requires space, so this is the spatial schema at the bottom, and one that doesn't, that requires them to pay attention to color. And so the way it works is the monkeys are trained to navigate in this open field, just like you saw in the very first video. But now the different targets that they'll collide with are worth different value of reward, either high, medium, or low, based on, in the color task, the color of the item, and based on, in the spatial task, based on its location in the field, even though that's unmarked to the animal. Okay, so let me show you kind of how this works. So this is the very first trial where the monkey was performing this color task. So this is a different monkey, Manchita. And if you can hear the beep, so she's got 2 beeps for the green, just 1 beep for the red. So she's just exploring the environment and getting different values of reward for different targets that she's colliding with. And so now she got one of the high rewards. So the 4 beeps go for the blue. And then just 14 trials later, here's what she does. So hopefully you can see she kind of looks around and then really sort of makes a beeline for the blue targets. The key to this task, I think, and for them learning so quickly, is that there's only 30 seconds per trial. And so they can't really forage for all the items each trial. So she has to be strategic. And she's learned really well. So she went for all the blue and then went for the medium ones and then ran out of time. And it's really fun to watch them do this task. There are some monkeys, we can tell that they're learning that there are 3 of each value, and also they get a real good sense of that 30 seconds, and they'll make decisions about if the next one that they're gonna go for is really far away. Even though it's high value, they might decide to go for a low value one that's closer by. So again, lots of interesting behavior. And then I'll just show you how the spatial task works. You can get a sense of this. So here, all the items, again, are the same color, and there are unmarked zones in this environment. So the monkey has to learn where the high-value items are. So those are worth 2. And the monkey starts each trial from a different location. So there she found the high reward ones. So now, again, this is just 14 trials later. She grabs the one that's really close by, and then you can see she just sort of orients herself to where the high value ones are, and again, quickly goes and gets those. There's, again, lots of interesting behavior that we're seeing with the eye movements in this task. So we're seeing changes in where they're looking and the items that they're focusing on as they're learning through different trials. Okay, so let me show you the behavior that now we've got across many sessions of this. So the monkeys are given new colors every day, and they're placed in a new virtual environment. So each day they have to do this new learning. But they can do it really quickly. So monkeys can learn this in about 10 trials, 10 to 20 trials. They can show nice discrimination between these different values, which is great because this is the kind of task now where we can record within a single session and try to track how that neural activity changes through learning. And so for these initial recordings, Sophie was using the Plexon S probes, and she's used multiple Plexon S probes, so she was able to record lots of neurons simultaneously. So for us, in these early experiments, 100, 120, this was lots of neurons. This is a huge improvement for monkeys. She was able to track the same neurons while the monkey learned the different tasks. And so the way she did this was she would give the monkey a block of the spatial task and then a block of the color task. And importantly, she was able to look at, again, the activity of the same neuron across these different tasks. So what I'm showing you here is the response of one neuron, one hippocampal neuron. This is a top-down view, and so the heat, it's sort of a heat map, so the warmer colors represent higher firing rate. And probably what you can see is even in the early phase of the session, there's a difference in activity for when the monkey's doing the spatial task versus the color task. This neuron is giving kind of a spatial representation for the spatial task, but not at all for the color task. And then if we look through the session, it even kind of sharpens up late in the session for the spatial task and really kind of quiets down when the monkey is doing the color task. So these are just alternating blocks. So again, there's nothing that we can say about this neuron being a place cell. It just happens to code for space when the monkey, when space is relevant, right? When the monkey is doing that task. Sophie's also been able now to train the monkeys to encode multiple value maps in a day. And so I'm going to show you an experiment where she was teaching them 2 color maps, and these are the colors that they used for that day, along with one new spatial map, recording from the same population of about 100, 120 neurons. And over here, this legend, sorry, I don't know if you can see my cursor, but the legend colors will sort of map on to the different spatial and color tasks that the monkey was doing. So early in learning, this is looking at the population dynamics, what she found was that there was really a lot of entanglement between these different representations. So the size, what she's looking at here is the 2 seconds prior to reaching the target, and they're projected into the space defined by the PCA analysis. And the size of the dots indicates the time as the monkey's entering or getting closer to the target. So the bigger dots are right when the monkey is getting to the target. So there was a little separation with the color map 1, but everything else was pretty entangled. And then if we go through, again, the middle part of the session, and by the late part of the session, there's a real separation in the population response to these different targets. So it suggests that really the population is able to sort of segregate into these different representations of these different targets. Now, interestingly, we didn't see any reuse of the subspaces of the different maps. And so I've sort of given you a triangle here to show you where the maps are. But each map and target appeared to occupy a different location in the population subspace. We have some collaborations going with Dave Friedman, who's been recording in frontal eye fields and posterior parietal cortex in a 2D version of this task. And they show that there's some evidence there's kind of a reuse in those regions, but not in the hippocampus. But again, these data, I think, are consistent with older findings from rodents, as well as a growing picture of studies across species that the hippocampal representation is constructed and modulated by task demands. So here I've listed a whole bunch of papers, and I need to add Weinan's paper to this. But what this is showing are all studies that have shown that the hippocampal representation and the activity correlates with and switches across the animal's goals. So in this experiment, they had the rodent performing 2 different tasks and showed that the activity was dramatically different just depending on what task the animal was doing. So again, supporting the idea that the hippocampal code is not fixed just based on allocentric space, but it really comprises this internal mental model. Okay, so an interim summary here before we switch gears to go to eye movements. What I've shown you is that in a very well-learned task, this delayed alternation task, hippocampal receptive fields chunk conceptually similar epochs, and the population activity really parcellates the salient task events. And also I showed you that through learning, a low-dimensional subspace can emerge rapidly, and it captures information about these discrete cognitive variables. And together, again, the idea is that the hippocampus constructs this internal model, and it really depends on the goals, the immediate goals. Just a sort of ad for future studies that are coming. So we have actually in the last few weeks started recording with NeuroPixels probes, and so these are hot off the rig. These are new data. But it's been amazing to see the kinds of activity that we can get with these NHP NeuroPixels probes, and the numbers are increasing. So instead of 100 to 120, now we're pushing a few hundred, I think, in each session. So I'm really excited about the new analyses that we're going to be able to do with these data sets. All right, so I'm going to switch to the second part, where I'm going to talk about eye movements and exploratory behaviors. And I think the use of eye movements to take in information is probably the most conspicuous example of exploratory behavior in the primate. And using eye movements as an index for cognition dates back to Alfred Yarbus from the 60s. And so here I'm showing you some scan paths that he collected in human subjects as he showed them these paintings and just changed the instructions that he gave them. So either to estimate the material circumstances of the family or examine the painting freely, you can see this dramatic shift in the eye movements. And so we had been using eye movements as a measure of memory in our studies in monkeys. And as we were doing these recordings, this was work with Nathan Killian when he was a graduate student in the lab, he saw that monkeys really showed this kind of interesting exploratory behavior as they were looking at these different pictures. And he was recording from the entorhinal cortex, trying to look for memory signals. And we sort of one day jokingly thought that, well, maybe these responses in terms of where the monkey is looking might be reflected in the activity of entorhinal cells. And so he went and did the analysis and surprisingly found grid-like activity that reflected where the monkey was looking on the screen. And so I'm just showing you one example down here at the bottom. And just to remind you all the beautiful kinds of spatial cells that have been identified in the rat, including place cells in the hippocampus, grid cells and head direction cells and border cells in the entorhinal cortex. And these are all through environmental exploration when the rat is moving around in space. We've been able to identify all these same kinds of representations, but based on where the monkey is looking on the screen. So this slide really encompasses about 10 years of work in the lab after that initial finding of grid cells to try to understand more about these kind of spatial representations. And there's been lots of other work, I think, in monkeys, but also in marmosets and humans, showing again these kind of strong spatially locked representations to eye movements. But the other thing that we noticed early on in the lab, and this was work by Mike Jutras in a couple of papers, where we found that the eye movements were not random in terms of their fixation durations, that they sort of occurred in a rhythmic way. And this is true both for monkeys and humans. So we tend to move our eyes about 4 or 5 times a second when we're just exploring scenes. And intriguingly, this happens to be kind of at the low end of the theta frequency, which has been really elusive in terms of trying to identify theta frequency in the primate hippocampus. So in this early work, Mike found that there was a reset of the theta frequency oscillation in the hippocampal LFP that was aligned to these saccades. So it suggested, so these data are showing that after the saccade, there's a more kind of alignment across different eye movements of the LFP compared to before, suggesting that what the saccade tends to do, or maybe the impact of the saccade, is to reset the activity to better process incoming information. So if that's true, then we wondered, would these saccades also structure single unit activity? We know from a lot of studies in rodents that hippocampal neurons tend to fire at particular phases of the hippocampal theta. So if we use the eye movements as kind of a proxy for the theta phase, would we see any relationship between the firing of individual neurons? And so this was work by Seth Koenig when he was a grad student in the lab. He was recording in the hippocampus while monkeys were doing this kind of free viewing of images. And here's an example of what he found. So at the top, you can see a raster. So each row is a different trial. And they're aligned to the time of the fixation start. Because the monkey is freely viewing, we're going to get variability in the duration of the fixations. So he's just sorted them. So the short fixations are at the bottom, and the long fixations are at the top. And the red line indicates the time of the next fixation. So hopefully what you can appreciate is that this neuron has a really reliable response. It fires, almost every time at a particular delay after the fixation onset. And when he looked at this across lots of neurons in the hippocampus, what he found was that the activity tends to tile the time of a fixation, which is about 200 milliseconds. And so you can see on the left a plot showing all the fixations, and on the right is cross-validated, so half of them but sorted by the other half, showing this really beautiful kind of rhythmic activity and also suggestive of kind of sequential activity after each saccade. And in thinking about this organization maybe related to a theta cycle, we were really motivated by a review article by Yuri Buzaki and David Tingley from several years ago, really making the point that in order to understand the hippocampus, you need to find the right alignment mechanism, right? And so what they're showing you in the top figure here is each color represents a different hippocampal neuron. And in the top, they're just aligning them based on distance down a linear track. Okay? And so you can see that these are place cells, so you would be able to decode the distance that the animal had moved down the track. In the second plot, what they're showing is the same data, but just now aligned to time, since the rat had moved down the track. So again, the rat moves at a pretty consistent pace, so you could decode time in the trial. But if you align the data relative to the theta cycles, that's when you see these really beautiful sequences. And this is due to the theta phase precession that we know occurs in the rat hippocampus. And so it's pictures like this that just make you go, okay, this is what we've got to do. We've got to find the right alignment tool for the hippocampus in order to really understand what it's doing. And so we were super excited then to kind of see what this activity looks like when we look at these saccade-related responses. So here I'm showing you 6 different neurons, and they're plotted in the same way that I showed you before. And to me, this kind of structure just jumps out and is something that I find really beautiful, and so I want to learn more about it. But I think it also, looking at these kind of plots, there are a couple other things that jump out to me. So one is that if we're just plotting the activity relative to the latency, kind of the time relative to fixation that this neuron has its highest firing rate, we're going to miss a lot of the structure. So each of these neurons, they have different latencies in terms of their peak response, but they're also showing these other really incredible patterns. So like, the neuron in the top right has this really interesting kind of decrease in activity and then a sharp increase, and then that activity kind of decreases. Some of them show kind of striped activity at some point. For most of these neurons, except for the one in the middle right, they have high firing rates. And based on the waveforms, they're all pretty narrow, so we think these are putative interneurons, but this is something that we want to follow up on. It feels, from the analyses, most of the neurons that show this activity seem like they're likely to be inhibitory interneurons. So we're just getting started with this, but I think it points to the idea that if you can find the right alignment, you start to see these really beautiful patterns in the activity. I'll throw this out there because we're still trying to find ways to characterize and quantify these kind of beautiful patterns. So if there's anybody who has great ideas, please contact me. I would love to chat about that. Okay, so I'll end here. I just want to conclude bringing you back to what I started with. I'm optimistic that our field is really starting to converge on a new phase of research with the promise of moving towards a unified view. I think we're getting beyond the debate about whether it's space or memory and really getting to, okay, what is the algorithm that this structure is involved in, and how does this lead to interesting behavior? And I think several exciting models have been proposed, and a lot of them by folks who often are at this meeting, including the Tolman-Eichenbaum machine, the idea of the hippocampus as a sequence generator, successor representation, and then most recently, I think the idea of afforded action progression within a task, which I think is kind of a similar flavor to the data that I've been showing today. And I think that each of these ideas include at some level the idea of a cognitive parsing of experience, which really can help us organize and prioritize relevant information, presumably making it easier to remember. So I think that's where the link would be to memory. And at the same time, our past memories influence how we parse new information, really shaping our perception and our understanding. So I want to leave you with one final idea, and that is that I think progress is also being made through comparative cross-species research. I've talked a lot about monkeys and rats, but I really also want to shout out to marmosets, bats, chickadees, mice, and even flies, and goldfish. I'm sure there are other structures that I should be including. And a critical aspect, I think, of our progress is really to take the behavior and species differences seriously, to think about what it is that's really appropriate for each of our model systems. And again, my overall point is that I think we should be looking at the internally generated dynamics rather than thinking about, for the hippocampus at least, rather than thinking about aligning to the external world. And I think when we're thinking about this, we often invoke the ideas of Kant, who talked about space and time as these sort of a priori intuitions that fits with a model of space and time as a kind of fixed box into which we place our representations. But instead, I think our data are leading us to consider the ideas of Leibniz, who came before Kant, and who wrote, I hold space to be something merely relative as time is. I hold it to be an order of coexistences as time is an order of successions. So time and space are considered as emergent properties of experience, and these hippocampal representations, as I've showed you today, can be stretched and compressed accordingly. All right, so I'll end there. These are thoughts we can ponder maybe over drinks at the party. But I want to thank first the monkeys that contributed to this work. They really are our partners in a lot of this work. And I'd also like to acknowledge the incredible team of scientists who I've had the privilege to work with. John Ruckman and Yoni Browning led all the work on the Y-maze task. And Sophie Landy has been leading the work on the color and space tasks during learning. And I'd like to thank our funding sources and our great team of collaborators as well. And thank you.
Thank you, Beth, for this wonderful talk. We have like a few minutes before the party, so question over there.
Hello, wonderful talk. I had a question about the earlier part showing the chunking in the hippocampus. It suggests that, I think of it as a collapsing of state space to states that are maybe space invariant but have some similar kind of conceptual meaning. And I guess I'm wondering if that presumes that has to be learned. And so I'm wondering if you see evidence that there's a higher dimensional representation that is more linked to veridical space that then becomes lower dimensional into these conceptual spaces with time in the monkey. And if so, where you think the signal for that comes from, because presumably it's not coming from symmetries or stabilities in the external world. And then if I can overload this question, lastly, if you see any behavioral evidence of what that means for the animal to have chunked space into these conceptually stable zones. Yeah, thank you.
Yeah, 3 great questions. That I don't have the answer to yet, but I'll tell you that these are all things that we'd love to work on. So the Y-maze task was, really well overtrained. But what we'd like to do is, with virtual reality, we've done some experiments where we put them in new environments. And we're working on analyzing those to see, the hypothesis would be that in a new environment, you might see this kind of more veridical representation that then once they figure out it's the same rule, that gets chunked down. We're trying to think about ideas, and if anybody has ideas they can share with me, of testing out the sort of what that chunking does to the animal. So one idea is that it might make it harder to appreciate things that are happening within that chunked time, right? You're insensitive to changes there. Or maybe if there is something new there, it gets a really strong representation because that's unexpected. And in terms of where the signals come from, I don't know. I mean, I think it depends on the task. I think there's some beautiful work from Joni Wallace showing interactions from the hippocampus and orbital frontal cortex that might be at play there. We're also interested in combined hippocampal entorhinal recordings to see what that sort of input, might be, how that might be affecting the chunking as well.
One final quick question over there.
Hey, thanks for your nice talk. I actually didn't know that place cells don't form in a goal-directed task where you don't need the hippocampus and where allocentric information is kind of irrelevant then. And I find it kind of elegant, actually. And I was wondering whether the place cell formation, you think, whether you looked at the evolution of the formation of place cell as a result of the evolution in behavioral policy in allocentric tasks, and whether they are kind of prior to improvement in behavior, or whether they're a result to a very kind of nailed down policy.
Yeah, I have not looked at that, but I think it's an interesting idea. I think one thing that we continue to struggle with is, I think there's consistency across many studies that have shown when space doesn't really matter, you don't see these place cells in monkeys. But there are studies in marmosets that show them, and trying to think about in rodents, how do we understand, does space really matter to them when they're just doing open field foraging? And so I think there's something that's missing there in trying to reconcile those different findings, and maybe having an evolutionary perspective could be interesting.
Thanks, I see.
Thank you. I think sadly we have to wrap up the session. Well, you can find Beth at the party. Thank you again for the wonderful talk, and thank you everyone.
Thank you.